All right, welcome everyone. Today we're going to break down the FDA's guidance on handling out-of-specification test results. This is an absolutely critical framework for anyone in the pharmaceutical space because it really gets to the heart of ensuring both scientific integrity and product quality.
So, here's our game plan. We'll start by understanding what an OOS result truly is, the signal. Then we'll walk step by step through the two phases of a proper investigation, hit the key decision points, and wrap up with the essential rules of engagement to make sure the whole process is compliant.
Okay, first up, the OOS signal. Before we get into the how, we need to establish the what, and just as importantly, the why. It's all about having the right mindset from the very beginning.
So, what exactly is an OOS result? Well, the guidance defines it really clearly. It's any test result that falls outside of those pre-established specifications or acceptance criteria. And this isn't just for the final product. This applies to everything, from the raw materials coming in the door all the way to the finished goods going out.
Now, this right here is probably the most important mindset shift in the entire guidance. An OOS result is not just a failure to be discarded. Think of it as a signal, a red flag. The absolute prime directive is that you must investigate to find the root cause, even if you know you're just going to reject the batch anyway. You can't just throw it out and pretend it didn't happen.
And that mindset leads us directly to the biggest pitfall, the number one thing this guidance is designed to prevent: testing into compliance. This is the practice of just retesting over and over again until you get a number you like. It's completely unscientific, it's a major red flag for regulators, and it totally misses the point of finding out what actually went wrong.
Okay, so with those core principles in mind, let's move into phase 1, the laboratory investigation. This is where the action starts, and it focuses on the immediate steps for the analyst and their supervisor.
The whole process kicks off right at the bench with the analyst. They have a very clear protocol. Stop. If there's an obvious error, say a spilled solution or a power flicker, they document it on the spot. What they don't do is knowingly continue a test that's already gone wrong just to, see what happens. And of course, they have to confirm the system was suitable to be used in the first place.
All right, from the analyst, the baton gets passed to the laboratory supervisor. Their job is to conduct a prompt and, very importantly, an objective assessment. This 7-point checklist is basically their roadmap. They're going to dig into everything: the raw data, the calculations, the instrument performance, to see if there's a clear, assignable cause right there in the lab. So after that initial deep dive in the lab, we arrive at what I like to call the critical decision gate. Everything that happens next hinges on the outcome of this phase 1 review.
This flowchart lays it out perfectly. You've basically got two paths. Path A is the simple one. You found a clear, undeniable lab error. Great. You invalidate the result, document everything, and the investigation can end right there. But if you can't find a clear lab error, you have to follow Path B. And that means you're escalating to a full-blown phase 2 investigation.
So what happens when there's no smoking gun in the lab? We enter phase 2, the full-scale investigation. This is where the scope gets a whole lot bigger and we start looking beyond the laboratory walls.
Now the investigation moves from the lab out onto the manufacturing floor. The quality unit takes the lead here, assembling a team to go through production records and the entire manufacturing process with a fine-tooth comb. They're looking for any potential issue, any deviation that could explain that OOS result.
Now, let's say that deep dive into production still doesn't reveal the root cause. At this point, you might consider doing some additional lab work, but it's critical to understand that this testing falls into two very distinct, very different categories: retesting and resampling.
And the difference between them is absolutely crucial. Retesting is when you analyze another portion of the original sample material you already have. Resampling, on the other hand, is a much bigger deal. That's when you go back to the batch and take a completely new sample. And you can only justify doing that if your investigation has proven that the original sampling process itself was flawed and unrepresentative.
All right, once you've gathered all the data from the production review and any extra testing, you've reached the final stage. It's time to conclude the investigation. And this is all about maintaining data integrity and making a final, defensible, and compliant decision.
And as you interpret the data, there are rules. The first big one: no averaging away your problems. It is completely inappropriate to average an OOS result with a bunch of passing results to make the final number look good. Why? Because you're hiding variability, and that variability might be the very thing telling you there's a problem. Every individual result has to be reported. Similarly, you have to be really careful with statistical outlier tests. Look, they can be useful for providing some context, but, and this is a big but, you cannot use a stat test as the only reason to invalidate a scientifically sound OOS result from a chemical test. It's there for information, not to give you an easy out.
Ultimately, this all leads to one of two final outcomes for the batch. If your investigation confirms that the OOS result is real and reflects the quality of the batch, then that batch must be rejected, period. If, however, the investigation is inconclusive, well, then the quality unit has a tough call to make, and they need to review all the data and always err on the side of caution.
And there's one more critical step we have to talk about. What if the batch in question has already been shipped out? If it's in distribution, you're required to submit a Field Alert Report, or a FAR, to the FDA. And the timeline here is incredibly tight, within 3 working days of discovering the problem.
We have covered a lot of ground. So let's take a moment to boil it all down to the most important actionable takeaways, the rules of engagement for any OSIS investigation.
This table is basically your cheat sheet. On the do side, always stop when you see an error, hold on to your samples and preparations, and report every single result. And on the don't side, never ever test into compliance, don't use outlier tests to just invalidate a result, and don't average away a failure. And that really brings us to the single most important idea, the whole philosophy behind this guidance. The goal here isn't to find a way to pass the batch. The goal is to find the truth, to determine the true quality of the product. That's how we ensure patient safety and maintain our scientific integrity.
Thank you so much for your attention. I hope this breakdown has been a helpful guide to navigating the FDA's critical guidance on OOS results.